Hello and welcome to another video of How to Science 123. Today we're going to be talking about enzymes. Sorry for the long wait. So this is just a quick introduction to enzyme. So an enzyme, you probably may have heard of these things before, but they're basically catalyzers of reactions. They help speed up reactions. So if we were to look at this free energy diagram, we see these reactants turning into products, but they have to get past this pretty big hump here, referred to as the activation energy. They abbreviated it as EA, which I always get confused. I always call it energy activation, but it's actually activation energy. And so these reactants have to get past this hub, like this sort of hump of energy in order to turn into products. And they go through this transition state and eventually turn into products. So enzymes help lower this activation energy and speed up this reaction because, the reactants have less of a hump to get by. The enzymes help speed up the reaction by reducing the amount of energy that's required for the reactants to get by this hump. And also, a quick reminder, enzymes cannot change delta G. The reaction will always be spontaneous if the reaction is spontaneous, so it'll always be less than zero. This is a quick introduction to enzymes. So we have what we refer to as the substrate here. I like to think of substrates as reactants within a reaction. Then we have the active site of an enzyme. And the substrate is going to bind to this active site and form what we call an enzyme-substrate complex. And this enzyme-substrate complex will eventually pump out products. So this is referred to as a complementary fit, and it's very important for enzyme specificity because we want to make sure that enzymes or specific substrates can bind to specific enzymes. So in this case, a specific substrate, has the right fit, and this enzyme has the right active site in order for that substrate to fit all nice and snug and create this enzyme substrate complex, making the enzyme ready to actually start catalyzing the reaction of the substrate and forming products. So this is kind of a cool little diagram that I found, which helps sort of summarize everything here. So we have these substrates which enter the active site, and we can see that the active site of this enzyme is very specific to these two substrates, and will form the enzyme substrate complex, and these are held together by weak interactions, specifically hydrogen bonding interactions, which are generally pretty weak. And that active site will lower the EA, or the activation energy, because of these substrates binding to the active sites, in the right way. And eventually those substrates are converted into products and the enzyme is freed up again to allow the next set of substrates to enter the active site. So there are specific enzyme conditions. Enzymes are basically proteins, and proteins need to survive in specific environments in order for them to work at optimal function. And so in this case, the optimal temperature for a typical human enzyme is 37 degrees Celsius, and 37 degrees Celsius is the, homeostatic temperature for a normal human body. And so we want to stay around that 30 to 40 degree um, temperature kind of range, or uh, obviously the optimal temperature for the human enzyme will start to fluctuate and the rate of reaction will be pretty bad. So that's why when we get sick, and our body temperature goes all the way up, we get really, really sick. And that's because a lot of the enzymes and a lot of our body's metabolic functions are kind of diminished and they're pretty slow because of this fever, this infection that sort of affected our body and made us feel very, very weak. And then this is kind of a cool comparison here. We have the optimal temperature for enzymes of thermophilic or heat-loving bacteria. These bacteria can survive in temperatures that are much, much hotter than 37 degrees Celsius, 75 degrees Celsius. So these bacteria are very heat-loving, as stated in their name. And so their enzymes are able to survive in much higher temperatures and, perform at a much higher rate of reactions. But then again, right, uh, if they were at a much colder temperature, for example, here, like around the optimal temperature for human enzymes, their rate of reaction would go down a lot more. They would not, it'd be very difficult for them for their enzymes to actually do anything at all. That's because they are thermophilic bacteria. They survive within those conditions. And then each enzyme can have an optimal pH. So Pepsin, for example, is a stomach enzyme. It's secreted during digestion. It's there to help break down peptide bonds and help, digest proteins. And then trypsin, sort of the same thing. It helps digest proteins, except it does that in the lumen of the intestine. And so this is important because pepsin has to be in a specific pH in order for it to be activated. So we have a pretty good note here. We have inactivated forms of enzymes. This is a pretty good introduction to that. Inactivated forms of enzymes are referred to as zymogens. And so the inactivated form of pepsin would be something like pepsinogen. And the inactivated form of trypsin would be trypsinogen. So if you see that ogen at the end of a specific enzyme, you would know that it is the inactivated form of that enzyme. So if the pH of the stomach here was around like 4, we would see pepsin would turn into pepsinogen because it's not activated. And that's pretty important because pepsin is a digestive enzyme, so it's there to break things down, and it digests proteins. And so if the pH of our stomach was, or if pepsin, if pepsin was inactivated, didn't have a specific pH to activate at, it would start digesting, our stomach because our stomach is obviously going to be lined with proteins, our body's going to be lined with proteins, and we would basically eat ourselves alive. Same with trypsin. And so the body's incorporated these zymogen or these inactivated forms of these proteins or these enzymes to activate during specific, moments within our digestive system. So when HCl enters the stomach, when gastrin secretes HCl from our stomach to basically begin digestion, pepsinogen will turn into pepsin and it will start breaking down those proteins because it recognizes that the pH in the stomach is at the optimal pH. It's at 2, it's a very low pH. And then trypsin is in the intestine, and you can see that its pH is a lot higher. And that's because trypsin is working within the intestine, and no HCl is going to be within the intestine. We want the intestine to be in a much more basic environment, less sort of around that, optimal pH of around 7, which is where a lot of that acid is going to be taken out of our intestine because we don't want that acid to be within our intestine. Our intestine is not specifically lined for that. And so trypsinogen will then turn into trypsin through the lumen of the intestine and it will do its thing in digesting proteins. We have cofactors and coenzymes. This is going to be pretty brief, but cofactors, for example, could be stuff like iron, or they're basically inorganic things, so like iron, zinc, and copper, so basically metal atoms. A good example of this is hemoglobin. It has iron as its cofactor, and iron's a heme group, and essentially iron helps oxygen bind reversibly to these hemoglobin molecules, so hemoglobin can actually transport oxygen throughout the blood. And without that iron molecule, or without that iron heme group, or that cofactor, the hemoglobin protein will struggle a lot in sort of reversibly binding oxygen and CO2 and transporting that around the body. Then we have organic cofactors, which we call coenzymes. They basically help the enzymes work better. I think you can refer to this as a key. When you're starting up a car, there needs to be a specific key that allows the car to start. And in this case, that could be the coenzyme. The coenzyme will bind to the specific site on the enzyme and essentially activate that enzyme so it can bind substrates and do its reactions and stuff. So now we have inhibitors. So there's two types of inhibitors. We have competitive inhibitors and then we have non-competitive inhibitors. Inhibitors are literally in their name. They just represent, stopping things. They stop things from happening. And in this case, they stop reactions from happening by making it so substrates cannot bind to the active sites of enzymes and catalyze reactions. So in this case, we're going to be talking about competitive inhibitors. Competitive inhibitors sort of match closely to the active site of an enzyme. And so they will sort of use that to their advantage by binding to the active site of an enzyme. Although they don't completely match with the enzyme, it's not their point. They're not trying to be the substrate. They're just trying to make it so the substrate cannot bind to the active site of the enzyme. And so now the enzyme cannot catalyze a reaction, cannot make products. And in this case, we have a non-competitive inhibitor, which sort of does the same thing as the competitive inhibitor and making it so that the enzyme or the substrate cannot bind to the enzyme. So in a non-competitive inhibitor, it does the exact same thing, except it doesn't bind to the active site of the enzyme. It binds to a site that's away from the active site, and it changes the active site of the enzyme, so now the substrate cannot bind to the active site of the enzyme. So that was a lot of words, but basically this non-competitive inhibitor is going to change the shape of the active site so the substrate can no longer bind. It doesn't have that snug fit, and the enzyme cannot form the enzyme-substrate complex and forego a reaction. And this is allosteric inhibition. Inhibition is not the right term. This should technically be regulation. But allosteric regulation is pretty important in the body because there are thousands of metabolic pathways that occur simultaneously in our body, and so cells need to regulate those metabolic pathways by switching on or off the genes that encode specific enzymes or by regulating the activity of existing enzymes. And so allosteric regulation occurs when a regulatory molecule binds to a protein on one site and affects the protein's function at another site. And this type of regulation may either inhibit or stimulate enzyme activity. This is important because our body is obviously always going to try to save as many resources as possible. If we have too much of something, our body's not going to waste time continuing to produce that same thing because it won't benefit us. So in those cases, our body's obviously going to activate or inactivate those enzymes to avoid the production of those products. So we have an allosteric enzyme here. It has, for example, 4 active sites in this case, and then it has a regulatory site on each of these 4 enzymes. So an activator will bind to the regulatory site of one of these enzymes, and it will allow the substrate to bind to the active site of the enzyme and create a reaction. And the opposite goes here. An inhibitor will bind to the same regulatory site on the enzyme and make it so that the enzyme is in a stabilized, inactivated form so that products can no longer be created, the substrate can no longer bind to the active site of the enzyme. It just changes the shape, similar to non-competitive inhibition. So in this case, we have a little metabolic pathway here, and this is the production of isoleucine. So the initial substrate, which is threonine, will bind to the active site of the enzyme. It will go through a number of metabolic pathways to eventually produce isoleucine. So the body's produced enough isoleucine in this case, and it's going to say isoleucine, hey, bind to the allosteric site here, and we can represent isoleucine as the inhibitor, and it will bind to the allosteric site or the regulatory site of this enzyme and change the shape of the active site of this enzyme. And so now that the active site has changed, threonine can no longer bind to the active site and continue to make isoleucine. And so once isoleucine is all used up by the cell, the isoleucine will unbind from this enzyme, it will unbind from the regulatory site. The active site is now available, and threonine can now bind to the enzyme and repeat the production of isoleucine because the body recognizes that there's not enough isoleucine in the body. And so this is an example of allosteric inhibition. And that's kind of all I have for enzymes. I hope you guys enjoyed the video. It's a pretty fun topic. Enzymes are definitely the most important proteins within our body. They do thousands and thousands of things, and they make thousands and thousands of things happen. And so I hope you guys enjoyed this video, and I'll be posting again soon. Thank you for watching.